as well as research experience and have worked in different central and state government universities prior to joining JNU, New Delhi. Sir is working in the area of biopharmaceuticals, process development, and toxicological studies of wastewater. Sir has various international collaborations, including Catholic University of Louvain, Belgium IEBT, Portugal IHE, Water Institute Netherlands, National Research Tomsk Polytechnic University, and East China University of Technology. So motivation is towards clean water for citizens of India. With this, I request Sir to please deliver his lecture. Please, Sir.
So, good afternoon, one and all, and thanks for introducing me, and thanks to Professor Yasha for inviting me to deliver a talk or to discuss some of the research which is going on in my laboratory. Though the topic of this Gyan course is more towards the translation of benchtop to bedside, like more on pharmaceuticals, how the things and therapies will be implemented to the patients. I am certainly going to talk on a same aspect, but with respect to the biomanufacturing, which is the present time the government's mandate also. Here I will talk two different cases based on Petri plate to bioreactor. How we can start if you are having some microorganisms from where you can isolate, how you can start working in laboratory, then how you can go for lab scale, then further how you can go for a scale up process, and what are the different challenges you can face. So it's a kind of whole story I will talk with this, which will start from Petri plate to the bioreactor. If you recall, just 2 days National Science Day we celebrated in our country, and the theme was that Science Day was like how we can empower the youth with respect to the science and technology, so that the country will involve the ecosystem for innovation, so that what we are talking about, the Viksit Bharat or developed nation in 2047, how we can achieve that. So it can only be achieved by with the help of youth. Our country is having more numbers, right? So here is also the all kind of the young generation and youth is present. So hope you would like to understand the current challenges with respect to the country and at the end at global level, and how you can solve those problems, right? So I will talk on the two different cases. The first is case with respect to the cancer. Like people are talking most of the time on antibiotics, how antibiotics is increasing into the water bodies, how antimicrobial resistance is generated. The present antibiotics or the first and second generation of antibiotics, they are not working on the patients. What happened? That means people are worried about the antimicrobial resistance which is generating. But nobody is now caring about the rise of cancer patients globally. Got my point? We are talking more on antibiotics. Why not to the cancer drugs? The worldwide the population is increasing. What happened if that drug is going to the water bodies and reach ultimately to the food chain? What kind of effects you can see? Maybe the number of cancer cases rises. You are not aware that this is the main cause behind that, but people are not aware about it. Okay, so we started working on that one aspect. What happened if like cancer drugs presence in the water bodies will be more? Okay, then the aquatic life will disturb, and ultimately if we take the fish and other components which are produced in water bodies, so that the cancer drug will reach directly to the unaffected person, and they are damaging in DNA replication, transcription, translation, many mechanisms they are changing. Okay, so here you can see the the kind of breast cancer and colorectal cancer and lung cancers numbers are increasing, though the data is old, but the present scenario is the worst than what you can see now. We are talking about the country like our India. Here you can see the data with respect to the various states. We are saying that the cancer is a disease in the urban areas. Like in earlier times, we used to say that diabetes is the disease of urbanized places. But nowadays, more persons you can find in the villages, in the rural areas. The same kind of story here you can also see. We said that in northeast region, they are not too much polluted. They are taking more healthy diets. They are more physically active than the places like Delhi. But what happened there also? The cancer cases are rising. In Rajasthan, Bihar, UP, you can see the sharp rise. So, like the cancer drug, when any person take as a chemotherapeutic agent, what happens? The 40 to 60% of the drug that come out through the urine. So, if there is a 1 ton consumption of cyclophosphamide, how much kg of cyclophosphamide, which is an anti-cancer drug, that can go to the water bodies? Globally, there is no monitoring system. Hardly few countries are having partial treatment system to remove that drug. So what is happening? So I started on that aspect, and the second aspect which I will cover, that is the obesity. That the country is, you can see here, if anybody is having more than 25.5 BMI, then person will considered as a obese. So that the second, I will also talk on that. So that means we need to lose the weight, but at the same time, if you look at here, the maximum risk is, like when we say the youth is responsible, we are targeting youth for Viksit Bharat, but what is happening? They are not normal person, they are going into the condition is going to be the obese. Even if you go on the gender wise, you can see how much men versus women are obese in nature. So what kind of treatment required? What are the drugs you can develop? So that will also, I will talk in my discussion. So we'll start with the first one. Hope you might know that molecule, colchicine. During like in the 10+2 times, that colchicine was chemical used to increase ploidy level. It's a plant alkaloid. Okay, but when you reach in the undergrades, master graduate, then you realize, oh, colchicine is a molecule which can be used as to arrest tubulin proteins. So that means it stopped cell division. It stopped tubulin polymerization. Okay, but at the same time, how you can produce colchicine, how you can use the colchicine, which is more potent and less toxic. Okay, so here it's a red lily, Gloriosa superba plant, from which you can extract colchicine. You can see the chemical structure where there is a 3-ring structure, but there are several methoxy groups are also there. This methoxy group, you can see, I can show you by laser. You can see these are the 1, 2, 3, 10 here also. Methoxy groups are present. But when this colchicine you used as an anti-cancer agent, so that means that structure is going to bind with the tubulin proteins. It's anti-tubulin, it stops tubulin polymerization. Okay, but when it bind with the tubulin, the potential of the binding of the colchicine is not that much effective, and that is also toxic also. It creates nausea, vomiting, and many other symptoms which any person, if took like chemotherapy, you can imagine baldness happens, nausea all the time, weakness, many symptoms come. So we identified that these groups are present. If we can remove one by one group, what happens? So we started removing these methoxy groups and started making trials, and we found that there is a specific methyl group methoxy group, which is a third carbon position, if we can remove that group, then the potency of that molecule increased, and as well as the toxicity is also very much decreased. So we started working on, can we identify some microorganisms, some enzymes which can do that, they remove that. So earlier, like nowadays, many chemical industries are producing, which is called 3-demethylated colchicine. You can see these are the different derivatives of colchicine, right? It's a colchicine, that is the 3-DMC, 3-demethylated colchicine. The thiocolchicine, which is thiocolchicoside. Okay, so you look at this molecule. So what happened? This, if we remove the third position group, then the toxicity is reduced, binding is increased. So we thought that, can we find out some microorganism which can do it? If we synthesize chemically, what happened when you are going to purify, because these are the chiral compounds, okay, when you isolate those compounds, it will be difficult and the cost will also increase. So we thought that, can we find out some new microorganisms which can do that? So hope you might heard many times in COVID time also, repurposing of drugs. Yes. Okay, many drugs repurposed. So that work was in year 2006-7, not in the 2020 or 2021. So that means the drug modified form of the drug requirement has been increased. So this is just, I'm showing the market size. Those are modified natural drugs, the market of those drugs are increasing. So if we can repurpose any of the drug, that can be useful. Okay, so on that basis, we started working on the molecules. So why we can, on what will be the parameter and what will be the criteria on which we can say that these colchicine derivatives is the best. So we have to, there should be some criteria. So we have selected the widely used some anti-tubulin molecules like benga alkalides, taxanes and colchicine also, and start forming some experiment. We did some experiment, we found that because colchicine binding strength is better than the benga and the taxanes. So we thought that we reached at a conclusion that if we can produce 3-demethylated colchicine, that can be useful for as a chemotherapeutic agent. Okay, so then we started doing several experiments, and this is the story of all kind of experiment which I performed. Like plant I showed you, from which we have extracted colchicine. Okay, then as I said, like we have to find out the microorganism or some enzymes which are responsible for the change in the colchicine structure. So we have seen that if a cancer patient, okay, administered the colchicine molecule, then that gene of the CYP3A4 was transformed into E. coli. Okay, then E. coli we started using for the lab scale level. Okay, so we'll show you data also how it goes. Recombinant, as I said, like the target was to develop recombinant was only 2 targets: how we can reduce the ergosterol content and how we can increase the yield. Yield means the percentage of conversion, transformation. Okay, so now go to the next slide. Here if you look at, I'm showing you the 3 possible experiment, different type of experiment we performed in Shake flask, in 5-liter bioreactor and 70-liter bioreactor. If you look at the total demethylated colchicine, here I'm saying that in all the experiment we use 7 gram per liter colchicine as a substrate in the medium. Okay, so in the sac flask you can see there is a conversion of out of like 7,000 milligram was there, so 3,800 was converted, so it's almost more than 50%. Okay, in reactor also, you know, like what kind of difference you can see in sac flask and reactor. When we say reactor, that means there are more controlled conditions which you cannot control in sac flask, like pH, like dissolved oxygen, like substrate concentration, product inhibition, which you cannot control. Okay, so in lab bioreactors you can easily control. So here if you look at in 5-liter reactor, the same yield was there, but the 70-liter it's jumped. Concentration, the conversion is increased due to like more controlled conditions. Okay, that this is I am talking about a Bacillus megatherium. All experiment performed Bacillus megatherium, and you look at the specific yield, then the specific productivity, that per gram, per liter, per hour, which is also important in case of bacterium. If you are using fungi, they took to grow longer time, in a week, 2 weeks. Okay, so here we are using bacterium, so there was no difficulty to grow. So the productivity you can also see it's increasing, and the specific yield milligram per gram of dry cell weight is also changed. So that is with respect to the Bacillus megatherium. But here what happens when we say that we started from the sec flask, then 5-liter, then 70-liter. How you can go from 5-liter to 70-liter? Do you heard scale up during your BTech? What is that? How you can do it? Suppose there are a 5-liter bioreactor and 70-liter bioreactor, and the diameter for both the bioreactor is absolutely completely different. So when in 5-liter reactor you are performing suppose 500 RPM, are you going to maintain same RPM for 70-liter? Not same, different. Whether they are increased or decreased? Decrease. So how you can calculate? What will be the criteria for a scale up? Okay, so we started with that experiment, and we took a impeller tip speed or the impeller tip velocity as a one of the parameter, so that we can say it is scaled up from 5-liter to 70-liter. Okay, so here you can see the RPM at 5-liter reactor level. Then we have calculated the tip velocity. Okay, the tip velocity formula is pi ni di. Impeller tip velocity is equal to pi ni di. Pi 3.17, ni means number of RPM, di is impeller diameter. Okay, so because the diameter of the vessel it changed, so that means the diameter of impeller will also change. Okay, so you take it that and calculate the tip velocity for a one specific size of reactor, here 5-liter. So this is the value. Now you want to constant this to maintain this much of RPM. Okay, so when you are going to the 70-liter reactor level, the number of RPM you can see they are reduced. So they are calculated on the basis of constant tip velocity. Otherwise you cannot say, how you can say for 200 to 87, what will be the criteria? Getting a point? So this is the way to scale up. When people say that they are doing experiment in 500 ml, now they started doing in 1 liter, and they said it is a scale up. What is the fun they have scaled up? What will be the criteria for scaling up? Because scale up is a completely a chemical engineering aspect. Okay, so there are several criteria. One of impeller tip velocity I am discussing. There is a power number is there. There is a KLa value is there. What is the volumetric oxygen transfer coefficient? Okay, so how you can keep constant? In 5-liter you are supplying 1 VVM air. In 50-liter or 5,000-liter, are you going to supply same volume or the half the volume? How you can calculate? What will be the criteria? So there are some scale up parameters on which you can calculate. So this is the way how you can going up and up with respect to the colchicine molecule. Here I published an article, and this was just to represent that how colchicine is converted by a recombinant system we developed, that is the E. coli system, which is converting into the 3-demethylated derivatives. Fine. So this is now I am talking about the recombinant system. So now the next slide, here you can see the problems with the recombinant system. What happened? Here you can see same images. You can see if we can immobilize these recombinant cells to get more conversion. So what happens? This whatever substrate is available outside the weed, outside environment, what is the diffusivity of that weed? How it can go inside? What will be the mass transfer rate? What kind of mass transfer limitation will be there? So in here you can see if the cell is inside the center of the weed, so if the substrate colchicine is going inside, it cannot. If the weed is tight, if weed is loose, so it depends on how much concentration of sodium alginate you are prepared. You might have prepared artificial seeds, or you prepared the whole yeast cell, you might have immobilized, or nowadays like nanomaterials are there, people are immobilizing enzymes on quantum dots on the nanomaterials. Okay, so but the limitation will always be there. Okay, so if limitation is there, whatever yield we are getting, or that yield is same or it is different. Okay, so we have to talk on this also with the next one. Here we have what we did, we developed a packed bed reactor system. See here the cylinder, we took the glass column, okay, prepared the beads with recombinant system, and now that enzyme what we called, which we cloned into it, that enzyme was P450BM3. You might have heard that. It's a protein of 450 wavelength, and with BM stand for Bacillus megaterium. C by P3 A4, that 3 will came from there. Okay, so you can look at here packed bed reactor system we developed. Here you can see this is the reactor where is substrate with colchicine was there, and with the help of peristaltic pump that is flowing through the downward side of the packed bed reactor system. Okay, and that colchicine is converting by maintaining the flow rate, because if the flow rate will be high, the material will go in and out quickly. So we have to check the reaction rate of the colchicine conversion. Accordingly, we have to set flow rate into the packed bed reactor system, so that when it goes out, the colchicine derivatives will go there. Then you can further extract it. Now that is the story of like upstream process. We have now scaled up and seen many things. Now comes to the downstream process. Like whenever you want to sell any of the product which is produced in labs, if you want to sell in regulatory markets of US and Europe, they have some specific requirement. How much microorganisms will be present, what kind of solvent will be present, what kind of residual solvent will be present. They have some specific guidelines. Okay, so that means if we are using chloroform is a polar solvent or non-polar solvent? Chloroform is a non-polar solvent. Methanol, ethanol are the polar solvent. Okay, so chloroform, hexane, and they are the non-polar solvent. Okay, so if you are using polar solvent, non-polar solvents are toxic. Like chloroform, if you are using for extraction, so if they are present even the residual amount, they are toxic. So if you want to sell the material into the regulatory market, the cost will be very, very less. That's all. The contamination will not buy. If you want to buy, you reduce the rate likewise. So we started thinking, can we develop a downstream process differently, which like chloroform, we can remove that chloroform, or we can reduce the volume during the extraction. Okay, so that this is the downstream process diagrammatic representation, which you might have learned during your BTech MTech experiment, how you can do it, crystallizer, column chromatograph, different time extraction, liquid extraction. So that was we established. So then after that we started seeing, like if we are putting colchicine into the medium and Bacillus megaterium was there, what is happening? So the cells will be in very much stress condition. Look at the transmission electron micrograph. This is the corner images, the yellow color marked. This is the simple one, control. Bacillus megaterium is there. If you look at by putting 5 gram, 7 gram, 10 gram colchicine, what you can see? Cell wall is almost finished. Hardly some plasma membranes are there. There is only few inclusion bodies in the control, but when the concentration increases, more inclusion bodies you can see. When you increase the any stress, then the cells will reserve the food for their survival. Where they reserve the food for survival? Inclusion bodies. So the number of inclusion body increased, which you can see. Okay. So then you can see when we also increase the concentration and want to see the scanning electron micrograph, here you can see in control the capsule kind of structure. Here you can see more sheath is formed. They want to save. Okay, they cannot do the reactions which you are expecting, which you want, colchicine say colchicine derivative conversion. Okay. So now what we did, we developed upstream, we developed downstream. Now the control, this is the packed bed reactor system which you diagrammatically I showed you. Here what practically we performed, which you can see, this is the glass column, controller is there. So by which we can have the experiment for colchicine conversion into that side. Okay. So now our starting was the stirrer tank system in batch mode. We reached at the packed bed reactor system, not fully packed bed, fluidized bed reactor system. Okay, it's a multi-phase kind of reactor system. Now in the downstream process, I said, if you had started using chloroform, which is toxic, then I again started by using some new statistical tools like MATLAB or the genetic algorithm toolboxes, and we have seen that by putting this design up experiment regression equation into the MATLAB equation, then we started using three different type of the solvents: chloroform, acetone, ethanol. That means one non-polar, two polar. So we are combining all the three to reduce the chloroform volume, so that the residual amount which will be present at the end in the powder of the derivative, so it will be reduced. And we have seen exactly what we were expecting. The concentration for chloroform and all these has been reduced very much, and acetone and ethanol concentration, we started using more polar solvent for the extraction purpose. So that was the first story with respect to the colchicine, how we started with the identification of microbe, we developed recombinant, we developed upstream, we developed downstream, we developed solvent system, we have seen the physiology, morphology of the microbe in terms of electron micrograph, we have scaled up the process from 5 liter to 70 liter with the specific to the impeller tip velocity. So that was the story of the whole for colchicine derivatives. Now if you have any question and with respect to the first one, we can discuss now, so that we'll start the second case, which was obesity. The colchicine has to be penetrate, it has to go inside the bead. Yes, because we prepared the different percentage of like 1%, 2%, 3% sodium alginate beads, and we have seen that the porosity of the each bead was different. We have seen under scanning micrograph, we just cut the beads, okay, then seen that the porosity was completely different. So here we started using 1% only. Here, yes. For recombinant, we were using 1.5% because cell size was less. E. coli size is lesser than the Bacillus megaterium. Bacillus megaterium, mega means it's the highest, the size of the bacterium is the maximum. So it is called Bacillus megaterium. Okay, so that was with respect to the now. As now another story was obesity. Okay, so like obesity is like everywhere is the problem. People started saying as body mass index, if it is this value, that value, then you become obese. Okay, there are some genetic problems by which persons are obese, but most of the time lifestyle problem, food things by which people becomes obese. How they becomes obese, what was, how I will talk, and how again the molecule you can develop and reach to the next level. So the epidemic, we recently gone from pandemic, so many persons, even though they are not aware about what is pandemic now, they called pandemic abhi gaya hai. So this is another kind of not pandemic but epidemic is there with respect to obesity. Okay, and our country is having very much affecting in the recent times, and the total population, we can say it's a 5%. Northern India obesity was most prevalent in the urban population, and the obesity rate was the lowest in rural population, male and female. That is just due to the more physical activity which we are not following in the urban cities or the urban conditions. Food habits.
So before you proceed to the next case, I had some questions regarding that. You mentioned that your starting point of the case one project was that certain cancer drugs were being excreted in the water bodies and then are being introduced to the environment that is not native to that, and it is harming. So have you thought on giving a thought that what colchicine and what its derivative would be doing in the environment in which, because ultimately that will be also excreted in the same environment. So how the environment will be affected from this colchicine and its derivative?
Very good question. But these are the drugs which are produced biologically. They are, most of them are biodegradable. We always not talk about all kind of chemicals which were present in the environment. We will talk about those chemicals which are not biodegradable, because environment have their own system. Because rivers are having, we said that they are having own system to regenerate. Okay, so that is the problem. The new molecule we started working on it, which is more effective though.
So is it same about the colchicine and its derivatives too?
No, it's a derivatives. It's for derivatives. Colchicine is also biodegradable. Okay, and that is the another story what I am talking about. I also worked on the removal of cancer drugs which are present or going into the water bodies. I am also working on it. So that is reason I am collating both the things, not collating both the things here, because that was related to the environment biotech, where I am working on removal of microplastic, and just to check, we developed Raman based sensor by just putting laser into the sealed water bottle, and we can say that much of microplastic present in the water bottle. Same in the case of cancer drugs, we developed a whole prototype for the removal of cancer drugs in the from cancer hospitals.
So one more question.
Please.
So you mentioned in the downstream process you use artificial neural networks, MATLAB and genetic algorithm in order to optimize the process so that these secondary contaminations are reduced in the process.
Yeah.
So can you elaborate more that how these certain computational tools help?
If I had some blackboard, then I can explain in more detail, but just wait, I am going back. Here I can, if I can, yeah. So what happens, suppose you are not knowing in for all these three ABC, we are saying that chloroform, acetone, ethanol, what kind of concentration you want to use. Either you have to go back on the previous literatures what others are using, or you have to generate your own. Okay, so like suppose for chloroform, acetone, ethanol, there is a 2:1:1 ratio. Okay, So there will be like, if you might have gone through this central composite design, maybe some PhD student who are in final year, because you are in first year, so that is you might not known about it. But there is a one book chapter in Whitaker, Stenbury Whitaker, Medium Engineering. There is chapter. So if you go and read that, they said that the central composite design can optimize any of the parameters at 3 levels or 5 levels. What is 3 levels? 3 levels means 0 means center point, means constant 0. Then they said -1, go that side, minus here, or +1, go that side, +1. That is the 3 level design. But when these people started working on it, they said, but 3 levels could not be possible. There are also errors were also there. Why not to go for another level which is called 5 level? So they said that -alpha, then +alpha. So they started multiplying this value with a specific multiple factor, that is 1.38 from that side, 1.38 from that side. So that will be more broader design. So that is a box. Okay, so box will having center point. These are the -1, +1, then again -alpha, +alpha. So wider range will be there for all 3. So by which you can generate a regression equation. Okay, regression equation means, that means these all 3, combination of these 3 individual, all what kind of effect they can be have on a specific response. Here our target was more recovery of the colchicine. Okay, so that was regression equation we generated. Then we started moving on the next side. This is called artificial neural network. Here we use the MATLAB, where we put it this as an input, regression equation as an input, and there are some layers which is based on the hidden Markova model, HMM model. Okay, so by which this neural network will be optimized, and here in the ANN the target will be to optimize very specific quantity. Here we are saying 2:1:1. It's not like that. Maybe 1.8, 1.3 is to 1.5. So it will be more accurate. So that is the reason we are going for the another level. Now even people are talking about hybrid approaches, where we are putting ANN and GA both. They are saying GA ANN model. Okay, so when we are going with this algorithm, again when we say genetic algorithm, there will be like, again suppose this is 0, -1, +1, 0, -1, +1. So when we are saying genetic, that means there will be some crossing over will be there, maybe there will be some mutation will be there. Then what kind of iterations you can perform by software, accordingly you can optimize the value. Got my point? So by this way we reach that after iterations that graph will appear. So on that graph we can say chloroform will be like suppose 50, more than 55, acetone almost 40, then ethanol will be more. So when we are using suppose 100 ml broth, 100 ml chloroform, so now it reduced to 100 ml broth, 50 ml chloroform, 30 ml acetone, and remaining will be ethanol. So it reduces the chloroform concentration. So whatever residual will be present, that will automatically reduce.
So I have a follow-up question from what we asked. As you told, like there are 3 or 5 levels in the central composite design. So first to give the example of center, that is 0, then -1, +1, both the directions, and then you said that there could be an error as well. So that error is -alpha and +alpha, the extension of the bracket?
We cannot say it's error, but it's like we are broadening the range. More precise. So like if suppose if you are using a NADH concentration into the medium, it's a costly chemical, very effective. Quantity was like suppose 1.23 microgram. Okay, but when you optimize, you reach that there is a 1.18 will be more effective. So how it 2 digit will come, or you can say 1.183. Getting point?
Yes.
Those are very precise chemicals which are having more impact on biosynthesis of the particular molecule. So those are has to be optimized. Now, even nowadays people said that there are PAT tools, PAT tools, you might have process analytical tools, and QBD approach, quality based design. When you are designing for any product, okay, then what kind of design will be there? It should be quality based. What kind of quality? Your response is product, na?
Product efficiency.
Recovery. So that should be based on QBD. And there are tools. These are the tools. Now I am saying that hybrid are also used. Many new tools are coming up.
And sir, which book chapter from Whitaker did you refer to?
I cannot recall number, but the chapter content is medium engineering, media engineering, and there you can see there are two type of design will be there. One is plaquette Berman, another is central composite design. Plaquette Berman design was used to optimize many variables to concentrate on 2-3 variables. Central composite design used to again go with only 2-3 to optimize the quantity. Like suppose you are having 12 variables, you don't know which is having more impact on your product, whatever response. So how you identify? So there is a design called plaquette Berman. You go with that, then you can optimize like 3-4, you reach at 3-4 variables. Okay, then further you can go with this central composite design to optimize the values. First you numbers, then values. You go deep and deep in. This is the like people are doing very much now. Maybe these new AI tools can do, if you put it the data, they will do it. So now to the next one, digestion of lipids. You already said that the obesity will be the problem by the food. So that means food means lipids. Lipids are going into the body. What is happening with the lipids? Here I am just showing you the diagrammatically. This is the triacyl glyceryls. They will converted by lipase into diacyl and monoacyl. Okay, and these will be further going to be deposited. How they will be deposited? I am showing you. Look at this. This is the mechanism. This is the intestinal lumen. Where is the marker? Yeah, here you can see triglyceride, they will convert by pancreatic lipase into monoacyl into fatty acid. Further they move towards plasma membrane, then they go to mucosal cell lines, triglyceride, apoprotein, and at the end they will deposit it into the adipose tissues. Okay, so that is the movement of lipid digestion. Fine. How I can reach that? I will work on that, this mechanism. So look at this image. You might heard about Achilles heels. Okay, the story आप लोगों को पता ही होगी. Okay, so we have seen that in this whole mechanism, the pancreatic lipase is the key point. If we hit on it, it will be work out. And we started thinking the different molecules, which is molecule which would be a competitive inhibitor of the pancreatic lipase. Okay, so we reached at a molecule which is called lipstatin. Lipstatin will आपको structure बताएंगे. So the lipstatin is a competitive inhibitor with pancreatic lipase. If lipid will present, lipstatin will present, pancreatic lipase will come, lipstatin will go and bind the active side of pancreatic lipase, so that pancreatic lipase could not digest with the lipid. So lipid will not digest, they will not be reached to the intestinal, and they can directly pass out. Okay, so that means we find out the key point to move on. Okay, so this is the story. Then now Then I seen what is the problem worldwide, is what is the scope of market. Being I'm an engineer, okay, so I'm talking about more on engineering side. Okay, so we have seen the what is the market of that drug if we could find out. So like our country is in mid region, and there will be lot of other countries also which are high risk with this obesity. Okay, then further we have seen that what are the different market players available in India. Those are interested to produce lipostatin, or they are in lipostatin production. I've seen that there is a one drug available in market in the trade name of early state, produced by Pfizer. Okay, and that is in early state there is a active ingredient was lipostatin. Okay, so we started working on it. There are many companies who are interested in lipostatin. Still our group is also struggling to produce lipostatin in high yield, what people call commercial scale, or because industry never satisfied, they always want more and more and more. How much? A bacterium is also having capability. Okay, so that is also not able to produce every time how much you are expecting. So these are the things, and now I will move on. So we find out again, we started from the scratch streptomyces, which is a well-known source to produce many of the bioactive molecules. We started also playing with that. We isolated some streptomyces, and by performing several lipase activity assays, we reached at a one bacterium streptomyces toxicity, which can be one of the best producer for lipostatin. So we started working on it, but at the same time we also seen that there is some problem with mycelial formation of the life cycle. Like they started with hyphae, they started with mycelium, then we go to the pellet. They will start forming pellet. If pellet will form, what will be the challenge? Okay, we start working on the. So here you can see this is streptomyces which we have isolated. Okay, so then we again like what is that? We started looking the sequence. We have gone through the whole genome sequence. We have sequenced that microbes. We have deposited into the gen bank. Then identify the species, and then we also find out the what kind of biosynthetic gene cluster was there. What are the different molecules that microbe can produce. Then developed process, many things we worked. You can see these are like lot of things here. You can see that article I have published. Leave it there. We'll again start from the scratch. We identified the streptomyces, start growing onto the plate, go to the sac flask, then extraction, then identification. We have seen that lipostatin is able to produce. Lipostatin is produced from the isolated microbe. Okay. Now the next target, what is the microbe? What is the species, genus? So we started with the go back here only. We started gene sequence, whole genome sequence, all story we did, then identified and reached Streptomyces toxicity as the microbe. So then upstream process optimization, again go to the different carbon nitrogen sources. We have checked different carbon nitrogen source to reduce the cost of the production. We tried many carbon nitrogen sources and growth medium. Again, many medium we have seen from the literature. We go for different plus minus. You can see some of them we have added something, some of them are negative things. So many things we did with upstream process. We also optimized seed medium because seed medium required more carbon rich, more nitrogen rich. So we provided more that so that the culture becomes young, and whenever we put into a next level in production medium, it can grow in a huge quantity. So many things we optimize. Now come to the story. So here you can see these are the colonies but you can very specific things. The colony and the center point are the colored and the outer of the colony will be the white in color. But after some time, it again becomes like after 100 hours, the whole colony becomes pinkish. Okay, that means there are some color compound. So now I am working on the proteins which are responsible for generation of this color. If we can knock out those proteins, or we can switch off these proteins by using light, because they might be light sensitive, because color is there. Okay, so if we can put red light, the mechanism will be stopped. If we put blue light, the process will start. So now we are working on that kind of process to produce that molecules. Here you can see this mycelia is produced. Mycelia is initial growth. But you know, the problem we faced again in the bacteria, Bacillus megaterium, you can recall the problem. Problem was ergosterol. Here the problem is the pellet formation. Look at the mycelium, and these are the pellets which are very dense at the center point. So if the substrate want to go inside, it is difficult. If product wants to come out, it is very, very difficult. So now we are working on how we can loosen this pellet. Here the pellet is live, and their pellet was sodium alginate wheat, that we can easily control by changing the percentage of sodium alginate. But here we cannot. So here we tried many experiment by putting talcum powder, by putting glass beads, and now we are playing with genetic engineering to stop this for pellet formation. We'll show you the data. These are the pellets which you can see on glass slides. These are the compound micrograph images. This is the whole mechanism behind the formation of pellet, like hyphae, then hyphae again starts coming, then center point, then mycelia, then the center point, then pellet. You can see the different stages. But again the next problem will come, which I again showed you the same place. Reasoning was the same, and this work also, the pellet. And if you might have gone through a book, PM Doran, Bioprocess Engineering, there was a chapter called Heterogeneous Mass Transfer. You can see this image, fixed law of mass transfer there. Okay, so whatever mass in, that cannot be 100% mass out. So that means there will be, this is the interface. So something will be go in with interface, then it goes to the system. So the concentration will always going to the down, down, down. So center point, there will be less medium component, so cells cannot grow. But if you recall the previous, there was a this wheat where the medium can pass on easily. Okay, then again, but there was another problem. You can say intraparticle adhesion or the interparticle attachment, whether the cells are immobilized on surface or more in center side. If they are more in center side, then this type of problem you can see. But if they are more on surface, then the problem will be less. Okay, so then I started again working on it. Here you can see these are the scanning micrograph of the pellet. Here you can see in the image C, there is a small this branches starts coming. It's EDO phase in the growth cycle. Growth cycle you might had log phase, lag phase, stationary phase, EDO phase, this and that. These are the branching will starts forming. But if you go in the next image, there the branching is more branching. So that means whatever void space available with this pellet, these branches will go inside and they will stop that space. So the pellet will be tight. When the pellet will be tight, again the problem starts. Okay, so now we reached at the conclusion, at a specific growth cycle phase, this branching will start problem for streptomyces. So what kind of proteins involved? Can we identify those proteins, go back in the reverse genetic engineering so that we can tweak those genes, knock in or knock out? Okay, so we have gone through this and observed that there are some proteins which are having less than 20 kilodalton in size, playing role into the pellet formation. And the conclusion came by a report of Streptomyces colicolor. They have identified some genes like DIV4A, SCY, PAR-A, FIL, the Philippe formation. Okay, so we reached on that level. There are proteins which are might be responsible. So we have gone for a sequencing of whole proteome, okay, with the less than 20 kilodalton in size, and on the basis of that we generate the whole interactome. What are the proteins which can be involved possibly? Here you can see there are the RPSB and tough proteins. Here you can say DNA K. These are the proteins which are responsible for the formation of cell membrane. Okay, that means if the proteins are more, the more cell membrane will be formed, more cells will be formed, more branches can be formed. So that means we are now going towards in a direction. If we could identify the structure of these proteins, then we can identify further, go for QRT-PCR, identify the genes and do the next level experiment. But at the same time, like for industries, they never prefer recombinant system. Industries, they never preferred system which are like more cumbersome, because there are few persons who are literate working in industry in production. Some of them are completely laymen. They don't understand this engineering and all. So I started doing some experiment. Can we change this pellet morphology by just changing the medium component? Okay, so here we performed several experiments like different carbon source, different nitrogen source, different inorganic salts, and by putting some parameters constant like size of pellet, biomass, viscosity, oxygen uptake rate. So you can see in the sucrose, in dextrose, in galactose, in lactose, the size of pellet been changed. In here, in nitrogen, you can also see, but you can see here in the inorganic salts, there is a specific observation in when we put ferrous sulfate into the medium, there will be no pellet form, because it is behaving like a chelation. So it is stopped forming even pellet, but it is not, pellet formation is also mandatory to produce lipostatin. We are not to say that we will not want to form pellet. We want to form pellet, but at the same time not tightly woven. Porous pellet. Okay, so by this we reached at, we also gone for different pH. We changed the pH, we changed the inoculum percentage, we changed the number of RPM. Many experiments we performed, and we have observed that yes, there can be change, but that change cannot be, we can say that it is 100% sure it could be happen, even though perform many experiment, triplicate, but we cannot say to industry you have to perform this. We want permanent solution. So we are working on some of the proteins which can be, like we can say they are 100% responsible for formation of the pellet. Okay, so this is about the upstream side. Then whenever you go to the industry scale level, they ask why you want to use carbon source like glucose, why you want to use yeast extract, why not to use oils. They are cheap sources, but here we have tried many fluor and oils, And that we found that very peculiar things in sesame oil. In the second last, the lipistatin was very high. We don't know why it happens. Then we started searching why it is happening. So we have seen that during the lipistatin synthesis, linoleic acid was required. Okay, that is, and in other oils, the linoleic acid was not present. Only the sesame oil was having where linoleic acid was present after identification by GC kiya humne, then we have seen that. Okay, so if we even like yield was just doubled. Okay, we have also checked by putting linoleic acid with coconut oil and mustard oil, the yield was also increased. So where is that? Why not to use sesame oil as a carbon nitrogen source? Okay, so these are the things on by which, because our industry always want, when you say in practice, so you have to go with what exactly they want, and they never want to spend more money. Okay.
So what is the control in that case?
Here, the normal one,
Glucose?
glucose.
Okay.
Glucose and nitrogen source yeast extract. Here again I am putting this sac flask and 5 liter reactor. We have not gone till the 70 liter here. Okay, so we are showing the 5 liter data. You can see the increase in the, you might have understand the batch and fed batch system. Okay, so here you can see the condition, dissolved oxygen and temperature by which you can get. Further we developed many mutants with the NTG, chemical mutagen, physical mutagen, gamma rays and all, and like many kind of experiment, because target was to increase the yield, because we like failed in changing the pellet pattern of streptomyces. So we tried another mutants and many experiments we performed. As I said, linoleic acid. So here we also performed to putting linoleic acid plus oleic acid in a specific ratio to see whether how much they are affecting the lipistatin yield. So many experiments we performed with different ratios. Here also you can see different concentration of L-leucine we performed, because whenever we could understand the synthesis of some molecules, we want that can we how we can increase, can we put this as a precursor into the medium so that the synthesis could be more, but many times it go with as you desired, but sometime it could not. So here is the whole story. This we are working on it. This we have optimized, produced 5 liter. I am showing you the data. We are working for 70 liter or next level to scale up the process for lipistatin. So this is the story of whole upstream process from scratch to the 5 liter bioreactor. Downstream process we've also optimized, but here again you can see we are using hexane, which is also a challenging solvent. Okay, so we are working on it, how we can stop using hexane, how we can optimize with different other solvents, but still we are working on it on that direction. This is the extraction procedure by which we have extracted this colchicine after like successful growth, then extracted, then the rotary vacuum operator. So this is the all story behind this. Downstream process is not that much we have optimized, we are still working on it, but we have changed the direction of the study. We started identifying the light regulated proteins. We started identifying these pellet forming genes. So the story moved in a more in genomics and proteomics direction, no more in now. We are not working on the downstream too, but whenever we could able to identify the problems with pellet and light regulated proteins, because we want to establish a system that, like if there is a glass reactor, you can put a specific light so that biomass will get increased? Yes. If biomass will get increased, if it is growth associated product will automatically increase. If we stop the light, specific light, the genes will be switch off. So that means we are interested now more in switch on, switch off mechanism of gene by using light. So now we change path with lipostatin. So that is the whole team for lipostatin work started by Puneet, followed by Khushbu. Indu also worked on a completely different aspect with respect to the lipostatin. She also, I have not shown that data. She put it a non-oxidative glycolytic pathway into the streptomyces to increase the number of acetyl coenzyme A. Like in most of the time, acetyl coenzyme A, 2 acetyl coenzyme A formed. Okay, but what happens when we put a additional non-oxidative glycolytic pathway? 1 acetyl coenzyme A increased, so 3 acetyl coenzyme A formed. When 3 acetyl coenzyme will form, the fatty acid synthesis will be more flux will be go, so then more lipostatin we can produce. So this we have another study where we have gone through like transcriptome study, transcriptomics, metabolomics of the streptomyces plane, the recombinant streptomyces where we put it the non-oxidative glycolytic pathway. So like more in that side. So C worked on that. Now third, fourth is working on more in metabolomics, light regulated proteins. So and 2 students working on like, one is already completed on the anti-cancer drug removal from the wastewater, and that he is working on the detection of microplastic in water bottle, sealed water bottle using Raman spectroscopy. So this Ansu is working on antifungal. We started some another molecule with antifungal production. Abhi the kind of black fungus problem came many persons. So we are working on the next molecule, which is pneumocondin, which is a like next generation antifungal molecule. So that is the our research funding. Obviously, we got national, international funding for that. Thank you so much for your all attention. Thanks to the Gyan coordinator for inviting me to share some of my work. Right. So now we can discuss and deliberate what I talked.
Sir, you talked about the pink color of the pellet, and then you said that you I guess get to exposed, or you exposed it to red light or something to see that it is responding to it or not in terms of group. So you talked about some light interaction with the pellet color. So sir, how do you get that idea if it will be light sensitive or not?
You know, like there are algae are already known for they are regulated by light. But you know, there are some bacterium also which have identified some light domains called love domain and bluff domain. LOV, light operated genes, and bluff means blue light operated, as likewise there is a full form. So these are the domains which are already identified in some bacterium. So we thought that this microbe is having color. If any microbe colony is having color, that means if we can put light, so then there would be either something will be increased or something will be decreased.
Some disturbance will happen. All pigments.
There are pigments, so it could be possible there will be some changes. So on that hypothesis we started doing that, and in my department there is a one person, Professor Sunil, who is working on that optogenetics area. So he's also producing some molecules from Chlamydomonas and study this eye wall ciliopathy, how the cilia will form in eye wall and how the light rhodopsin will regulate that cilia, and they are mimicking that into the algae.